Hello everyone, welcome to Biology in Minutes. Today, we'll be talking about protein synthesis part 2. Part 1 is in the description. Make sure to check that out. So first, let me just restate the process of protein synthesis. It starts out with transcription, then it goes on to post-transcription, and then translation. I have covered transcription and post-transcription in part 1. Right now, I will be covering translation. So after post-transcription, we got a mature RNA. And now how do we turn that into a protein? So before we talk about that, we need to understand what transfer RNA is, or tRNA. Transfer RNA is a single RNA strand with around 80 nucleotides, so relatively small. It transfers an amino acid to a growing polypeptide strand. It also has an anticodon, which is a nucleotide triplet. In this diagram here, you can see the structure of tRNA. On top, there is an amino acid, which shows that the tRNA is actively carrying around the amino acid. On the bottom is the anticodon. So where does translation take place? It takes place in the ribosome. A ribosome has a large subunit and a small subunit. The mature RNA strand, after post-transcription, comes in between those two subunits. An important word to understand is codon. A codon is 3 nucleotides that code for an amino acid. This circle chart here shows how different codons code for different amino acids. Remember, 3 nucleotides code for 1 amino acid, 3 nucleotides or 1 codon. When you look at this chart, the codon AUG codes for the amino acid methionine. Now that we established codons and that codons code for different amino acids, we have to ask, who brings the amino acid? The answer is tRNA or transfer RNA. This diagram here shows the overall process of translation. This arrow here demonstrates the codon, which is UUG. The tRNA here recognizes the codon and brings over the correct amino acids. How does the tRNA recognize the codon and bring over the corresponding amino acid? Let's look at some of the rules for the codon and the anticodon. If you have an A, which is adenine, it corresponds to U, which is uracil. If you have a U, it corresponds to A. If you have a G, which is guanine, it corresponds to a C, which is cytosine. If you have a C, it corresponds to a G. Here is the codon. Can you try to find the correct anticodon sequence? Pause the video and try it out for yourself. Here are the answers. Now going back to this diagram, the tRNA recognizes the codon and brings over the amino acid. Over here, we see that the amino acids are accumulating. After the RNA finishes translating, the amino acids form a protein. Thank you for watching.